Does this change things? Or does oh, okay. Okay, thank you very much. So I'm going to talk about Ebola viruses and in particular what makes them pathogenic in humans. So I think everyone will be aware of the outbreak that occurred a couple of years ago. This was the West African outbreak of 2013-2016, and this was the largest outbreak of Ebola virus that we've ever seen. So there are almost 30,000 cases and more than 11,000 deaths just from this outbreak alone. And it primarily affected West Africa, with Sierra Leone, Guinea, and Liberia the worst affected. And after it was contained mostly in 2014, we did see multiple flare-ups in cases in 2015 and 2016, and we know that the virus can persist in immune-privileged sites and that it's present in many bodily fluids. So this outbreak was caused by the Ebola virus species, formerly known as the Zaire species, and this is one of the 4 species that are pathogenic in humans. So the other ones are the Sudan, the Typhorus, and the Bundibugyo, and these 4 cause Ebola virus disease, which is the hemorrhagic fever, which in some outbreaks has fatality rates of 90%. And in brackets here, these are all of the years where outbreaks have occurred, so this is the most frequent cause, but the others have caused outbreaks too. And including for the Ebola species, there is a current outbreak in the Democratic Republic of the Congo. But there is a 5th species of Ebola virus, which is the Reston species. And this has never been observed to cause disease in humans. So we know that it can infect humans, but it doesn't seem to cause disease. However, it does infect and cause disease in non-human primates. So we wanted to compare these 2 groups to reveal what's the molecular determinant of this pathogenicity. So this is what the Ebola virus genome looks like. It's a negative single-stranded RNA virus in the Filoviridae family. It's around 18 to 19 kb genome size with only 7 proteins, so it's quite nice to work with. And here are the proteins here. So this is the glycoprotein, GP. This is the only surface protein responsible for attaching to host cells. And then we have 4 VP proteins and NP. Many of these have multiple different functions. And then we have the polymerase protein, L. So from the outbreak in West Africa, we had many more sequences than we've ever had before for Ebola viruses. So there was lots of sequencing that was done in field using MinION sequences. And so again, we wanted to compare all of the human pathogenic and the non-human pathogenic species. And our approach was to define specificity determining positions, or SDPs. And these are positions in sequence alignments that are differentially conserved. So we have our 2 groups. One group is the pathogenic and the other is the non-pathogenic. And we're looking for positions like this one with the red arrow, where there's conservation within this group of a valine, and there's conservation within this group of a lysine, but it's not the same conservation between groups. So this position here, where it's alanine all the way across, does not count. And this one, where there's variation even within groups, also doesn't count. So these are the prime candidates to be determining this difference. So we used this approach, and we applied it to all 7 of these proteins. And originally, we did this in 2015. So this was during the outbreak when it was still going on. And we had 200 genome sequences spread across the different species. And what we wanted to do was, since then, there have been many more sequences that have been released, and we wanted to update this analysis. And we now did this with more than 1,400 genomes, where we've had increases in the number of samples for most of the species. So can more sequences allow us to refine the SDP set and also improve our understanding of Ebola virus pathogenicity? So in 2015, we defined 180 of these SDPs at the sequence level, and with our updated analysis with more genomes, we have 166. So it's a net reduction of 14 SDPs, but we actually lost 34 and gained 20. So these are positions, the ones that we've lost, when we had 200 genomes, they look to be differentially conserved. But when we look with more genomes, we actually see there's much more variation. And for the ones that we gained with a small number of genomes, they look to be variable, but actually they're quite conserved. And on a per gene basis, in red here, this is the number with the small number of genomes, and in black is the updated number. For NP, GP, and L, we had net reductions, and for all of the VP proteins, we had net gains. So just two of those in detail. This is just a representation of the sequence alignment. And in red, these are positions, SDPs that we lost with more genomes. Grey are ones that we had both analyses. And blue are ones that we gained when we used more genomes. So you can see for L, we mostly lost SDPs, and we gained 2 more. But for VP40, we didn't lose any SDPs, and we gained 7 SDPs. And we only had 9 to start with, so it's quite a substantial increase. So what we propose for this set is that a subset of these SDPs will explain the differences in the pathogenicity of the species. So the next step after the sequence analysis was to move to structures. So we extracted all Ebolavirus structures from the PDB, and where there weren't any available, we modeled them with Fire2. And then we mapped our SDPs to structure. So previously we had 43 SDPs that we were able to map, and now we have 82 SDPs that we were able to map to structure. Most of this increase in the number that we were able to map onto structure came from our ability to model L, which is the polymerase protein, but the actual functional regions of L are still very much ill-defined, and so what we can infer from these SDPs is very little. But we did manage to map SDPs to other proteins too. So for how many of our SDPs could we actually find a likely functional impact for? And the answer was relatively few. So in 2015, we had 8 that we thought would be having a likely functional impact on the proteins. And in 2018, we have 9, including all of the original 8, just plus 1 more. So 2 of these we think are having likely stability effects on the proteins, and 7 of these we think are affecting interfaces with other proteins. So let's look at some examples. So this is VP40. This is one of the multifunctional proteins of Ebolavirus. But when it forms this oxamer, its role is in viral transcription, where it binds RNA. And we found this SDP, shown in red here, which is right in the interface of this octamer, where it forms the octamer. All of the pathogenic species have a proline, which forms this tight turn in the structure. And all of the non-pathogenic species, the restin, have a threonine here. Obviously, and we think that the loss of this tight turn and the introduction of the threonine side chain is likely going to have a difference in the stability of the octamer and the way that the octamer forms. Another Ebolavirus protein is VP24, and the main role of VP24 is inhibition of the host immune response. So physiologically, there are these 2 human proteins, karyofurin and STAT1, and they bind, and karyofurin helps STAT1 accumulate in the nucleus, where it will upregulate interferon signaling. So this is important to set the host into a state of defense against the virus. VP24 is able to bind to karyofurin and then prevent this accumulation and prevent interferon signaling, which is important for the virus, obviously, to have a successful infection. So one of the SDPs that we found in VP24 is shown here. So this is, in grey, this is VP24, and in blue, this is the human karyofurin. And all of the pathogenic species, this is the SDP here, have a threonine at this position, which is able to hydrogen bond with D48. So it links these 2 regions of the protein. All of the non-pathogenic species have an alanine at this position where they can no longer form this bond, and we think this will be having a stability effect on VP24, and also likely changing the flexibility of VP24 because these 2 regions are now no longer linked in the protein. Also in VP24, we found 4 SDPs, shown here, that are right in the interface between VP24 and human karyofurin alpha. And we think that these are likely to be changing the way that VP24 is able to bind to human karyofurin and changing its ability to downregulate interferon signaling. And now there's actually been some experimental evidence for what we saw here. So using reporter assays, this is studied by Gito et al. They showed that the restin virus, the VP24 of the restin virus, is less able to downregulate interferon signaling than the other 4 species. So we have these 4 species here, which get to about 90% reduction compared to the control, whereas the restin virus can only downregulate interferon signaling to about 70% of the control. And also VP24 has been implicated in a lot of adaptation studies where we take Ebola viruses that are not pathogenic in rodents and try and adapt them to the new host. And you see a lot of changes in the interface of VP24. So from our set of STPs that we had at the sequence level, how many do we actually see a functional impact for? And the answer is relatively few. And why is this important? Well, restin virus actually circulates in pig populations in the Philippines and in China. And so it's possible that restin could become human pathogenic, and this would actually only require a few mutations. And obviously, if this did happen, it could cross over into humans, which would represent a severe public health problem. So in summary, we've identified various differences between the Ebola virus species that may explain differences in pathogenicity. Our previously identified functional changes were retained in our new analysis, and this suggests that our approach is fairly robust. So we saw very similar results using 200 genomes and using more than 1,400 genomes. Again, we emphasize the importance of VP24, and experimental studies are starting to see the same thing. And we also think that it would only require a few mutations for restin to become pathogenic in humans. So with that, I'd just like to thank Mark, my supervisor, and also Martin, Stuart, Marina, and Miguel, who worked on this project. And thank you for listening. Possibly. So S3Det, the algorithm, does do some of that, but also in these, if you actually look in the alignments, there's almost 100% conservation most of the time for these STPs, so they're very well conserved within the groups, but possibly.
Do they have the structure for the curfilm for the other non-primate species that are affected by the non-pathogenic?
That are affected by restin?
Yeah.
I'm not sure. We have obviously the sequences of those, but I'm not sure if there's a structure.
Because it'd be interesting to model if they bind better to the non-human primates.
Yeah, the experimental evidence seems to suggest that they bind at least differently.
Yeah.
Do you think some of the SDPs might be non-functional but implicated in the interaction with host proteins? Have you investigated that?
Non-functional to the Ebola virus?
To the virus, yeah.
Oh, yeah. Yeah, possibly. It's definitely something. Like, for example, these ones in VP24 are responsible for the interaction with the host, I guess.
But other interactions, maybe with the immune system of the host?
Yeah, I mean, so VP24 doesn't just interact with caryofurin. Well, it interacts with a few different caryofurin, but it also directly interacts with STAT1 as well, which is downstream of caryofurin.
So how much is the conservation between different species? I mean, the sequence similarity for different Ebola strain?
It's pretty high. The figures off the top of my head, I don't know. The the pathogenic species are more similar to each other than they are to the non-pathogenic species. But even so, the sequence similarity is still high, especially for the coding regions.
So currently, you are looking at any way to do you think if there are some more information, if you consider like a correlated mutations of two positions or something? Well, currently, you know, if a position is not well conserved, you throw that position away from the analysis. But if you look at the other positions which are mutating, not conserved across, but have some correlation in the mutations, will you be able to do?
So you sort of mutually exclusive
Yeah,
positions that, we haven't looked at that, but that would be really interesting, yeah. Yeah.